May LIFE RCN Seminar. So the LIFE RCN is a network of astrobiologists who focus on the coevolution of Earth and life. We study ways to advance this science and its implications for our understanding of life on other worlds. So everyone will be muted for the entirety of the seminar. I would like your meeting ID to match your real name. You can ask questions by typing your question into the chat. So after each speaker finishes their talk, there'll be time for questions. To ensure that we hear from as many participants as possible, we might have to skip some of the questions, especially if there's multiple from the same individual. And all these expectations are, all the various expectations I'm going to go over are expected to be maintained in the Zoom chat, Q&A sessions, and in the comments on video recordings of the seminars posted on the Life RCN YouTube channel. So the Life RCN Seminar Series is dedicated to facilitating community participation in a respectful environment. We don't tolerate harassment of any kind. We ask that the questions and discussion are constructive and respectful. And the seminar organizers and moderators can stop participants from displaying disrespectful attitudes or making inappropriate comments. So if you notice misconduct or if you have any questions or suggestions, please email the Life RCN co-leads Frank Rosenzweig or Mary Drosser. So I want to give a brief plug to our upcoming session at the AbSciCon conference. So Alta Howells will be leading this, and this will be a really cool panel talking about how we can define unifying principles for detecting life. We've got 4 experienced panelists, and this session will be taking place on May 21st, 12:45 to 1:45 as part of AbSciCon 2026. All right, so we've got 2 speakers today. I'm really excited about today's session. It's going to be on evolution of multicellularity. So first talk is going to be by Dr. Núria Ros-Rocher. She's a CNRS research associate at the Institut Pasteur. She does really cool work looking at the evolution of animal multicellularity and identifying environmental cues that might drive this transition to multicellular development. And I'll be giving the second talk. My name's Dinah Davison. I'm a postdoctoral fellow at Augusta University. And I'll be talking about how Volvocene cellular differentiation can evolve rapidly and repeatedly through changes in plastic responses to the environment. All right, so I'll go ahead and stop sharing my screen, and Núria, take it away.
All right. Can you hear me well?
Yes.
Perfect. Amazing. Well, welcome everyone and good morning again. And thanks so much for this amazing opportunity. So as Dinah was saying, my main research focus is on trying to understand how the environment influences unicellular to multicellular transitions using close animal relatives as emerging model systems. And the main question of our lab is to try to understand how such complexity of body plans and morphogenetic programs evolved in life from a single-celled ancestor. And fortunately, nowadays, the way that we have to approach this is to use a comparative framework using our closest living relatives. So this is just a simple cladogram depicting animals, and then the closest lineages that have like very weird names. So you don't need to remember all of them, but basically we have a group of nanoflagellates or amoebas or amoeboflagellates that can be found in any kind of environment. Like they can be free living in aquatic environments, they can be on soils, they can be parasites of animals or other invertebrates, for example, or other like even fungi or plants. And the cool thing is that their genomes encode many genes that were previously thought to be animal-specific and that have key functions for multicellularity. The other cool thing is that these close relatives, despite being very simple apparently, they can respond to the environment and also form transient facultative multicellular forms that look like this. So for example, here we have a group of guanoflagellates. Here we have a group of amoebae that belong to this Philasterian group. We have here like two other representative of other relatives that are mostly parasites of animals. And mostly across the tree of life, there are two main ways to become multicellular. So we can transition from one cell to multiple cells by clonal division. So this is typically like serial rounds of cell division from a single founder cell that produces a clonal organism or a clonal colony. And this is typically seen in complex lineages, so those exhibiting three-dimensional morphogenesis and more complex body plans like animals, plants, algae, and fungi, for example. And normally they go through this typical single-cell bottleneck during development. But this can also be seen in simpler forms. And indeed, across the close relatives of animals, we have guanoflagellates and other relatives that, upon serial rounds of cell division, as you can see here in the video, they can form these transient colonies. And as I mentioned, this has this potential for complex forms. The other way to become multicellular is aggregation. So this comes where independent cells, sometimes from different genetic backgrounds, they can form a transient cluster or colony of multicellular cells. And this has been also seen in some of these groups of closely related lineages to animals. For example, we have a representative here of the Philasterians, in which you can see that they can form these cute clumps of cells. But there are more examples across the tree of life, such as, for example, even in prokaryotes, and then in other lineages closely related to animals, and other, like, for example, excavates as well, and rhizarians, etc. So if you compare both forms of becoming multicellular, there are different trade-offs. So, oops, can you see my screen? Yeah, okay, disappeared. So there are like different trade-offs. So for example, the more complex lineages, they can like have this potential for complex multicellularity, but then the more simpler lineages, they have these trade-offs because they can emerge, the genetic conflict can emerge. And that's why the question is, why aggregate, or why forming these more structures that are susceptible to genetic conflict? Well, the main reason is that they can actually confer a fast response to respond to suddenly like changing, fluctuating environments, and they actually have evolved, like most of these lineages, they have evolved mechanisms to recognize different cells from each other, and that's the term of kin recognition. So the story I'm going to tell you about today, it's our work that I've been doing during my past postdoctoral research in the lab of Thibaut Brunet, in which we focus on the group of organisms called guanoflagellates, that they have this stereotypical body plan in which you can see a cell body, an apical flagellum, and a collar of actin-filled microvilli that they use to swim and to capture bacteria that they use for feeding. We know very little about which cues trigger these unicellular to multicellular transitions in these lineages. We know that in the case of these organisms, Salpingoeca rosetta, which is a guanoflagellate, there are certain bacterial sulfonolipids that are actually in co-culture or present in the bacteria from the environment in which this organism was isolated from, that trigger clonal division. And this actually contrasts with other forms, as for example, aggregation in dictyostelids, for example. So these are the social amoeba. That is, for instance, the lack of nutrients that trigger this behavior. But we know many other cues that actually influence these unicellular to multicellular transitions in other lineages. So for example, abiotic cues or interspecies interactions. And the story I'm going to tell you about today is about this organism called Guanoeca flexa, which was recently discovered by Thibaut Brunet and colleagues in little splash pools filled out with seawater. So it's a free-living organism. And this is how it looks like. They form these beautiful cup-shaped colonies made up of a few hundreds of cells that exhibit this collective behavior and can invert their curvature upon different stimuli. It's actually becoming an emerging organism in our community because since its original description, we have stable monoxenic cultures. So this is co-cultures of these species with their prey bacteria. It can grow in large numbers in the lab, and we had at the time available protocols for cryopreservation and a transcriptome. And in our study, we are providing the genome and also different strains that we isolated, and we hope that in the near future, we can provide the genetic tools for functional assays as well as some other omic data sets. As I mentioned, this organism exhibits this collective behavior in which all the cells, they can completely invert their curvature inside out, not only upon light to dark stimuli, but also upon some chemical stimuli, like, for example, nitric oxide, as well as, for example, temperature, flow, or touch. As you can see here, the poor colony that being bothered by the pipette tip. Nevertheless, when I joined the lab, still there was a fundamental question that need to be solved, which is how and why these multicellular colonies that we call sheets, because they form like this monolayer of cells, are formed. And for this, we suspected that most likely, since it's a quanoflagellate and the other sister species, Salpingoeca rosetta, was forming colonies by clonal division, was that they could form colonies clonally. And for this, we did a very simple experiment. We isolated a single cell and monitored it over the course of several hours. And we could see that upon division, cells remain together, attached by these microvilli, the contacts at the collar, at the tip, purely by cell division. Nevertheless, here you can see a mid-sized colony. You will see a time lapse, and I want you to focus on these two cells. You will see that they can divide perfectly here and here. But sometimes, over the course of random cultures, we could see, here you can see like a doublet of random cells joining the colony and forming part of the colony by a process that remained aggregation. So then we wanted to ask whether this potentially mixed strategy of clonality and aggregation would be possible, and if cells were actually capable of forming colonies purely by aggregation. And for this, we did a very simple experiment in which we mechanically dissociated a culture containing colonies into single cells, and then we stained the two different populations of cells using live dyes and mixed them to see the outcome. So if we were to have like a single color, that would be a readout of clonality. But if we had like mixed or chimeric colonies, that would be a readout that they were produced by aggregation. And indeed, here you can see an Iris-scan 3D reconstruction of a colony after 24 hours of being mixed. We can see that it's completely a result of aggregation. And we actually tested that it's indeed independent of cell division, because regardless of whether we block cell proliferation or not, colonies that they can still be formed. And that indeed they maintain the stereotypical morphology of being a monolayer, the cells maintaining the contacts, and it's a pretty fast response, given that in the course of 24 hours we can obtain colonies as big as few hundreds of cells compared to what we would obtain by clonal division alone, that would be like few dozens of cells, assuming maximum and synchronized proliferation. If we compare clonal sheets and aggregative sheets in terms of morphology, they are completely comparable. So there's absolutely no difference in the way they organize the colonies, in the colony size, and also in their efficiency of preying or feeding on bacteria. So we quantified the amount of bacteria that they can trap using the flow that they generate with the flagellum. And you can see that comparing multicellular colonies in the aggregative state or clonal state, they are completely indistinguishable. And indeed, they still feed more efficiently compared to when they are like just single cells. So in this case, multicellularity confers an advantage of more efficient feeding, and then aggregation confers an advantage of establishing multicellularity faster. Moreover, if we also compare aggregative sheets versus clonal sheets, and we examine whether they can exhibit this collective behavior, you can see that upon light stimulation, the colony that is chimeric will indeed invert the curvature and contract, responding to the light stimuli, meaning that they are the aggregative sheets or the aggregative colonies, they are also comparable in terms of morphology. So given this mixed strategy in which Guanoeca flexa, this organism, can decide between being purely clonal, having a mixed strategy, or being purely aggregative, we wondered whether the natural environment in which this organism live plays a role in influencing transitions into and out multicellularity and the distinct modes of multicellularity. So for this, we were very lucky because we were able to come back to its original sampling site, and this is, it looks like this. So this is indeed a very harsh environment, even though it's located in the Caribbean. This is in the Caribbean island of Curaçao. And the environment in which they live are like these little splash pools that can be filled out with seawater, with rain, or by waves. So they experience these transient cycles of evaporation and refilling. So even though it's the Caribbean, and we can think about the Caribbean about a very relaxed and vacation time, it's actually a pretty harsh and highly fluctuating environment for the organism. So we wanted to actually try to correlate whether any parameter in which the organism lives actually is correlated to the multicellular behavior of C. flexa. So for this, we performed 2 independent expeditions in which we sampled more than 400 splash pools, and we tried to correlate any of these parameters by measuring, for example, temperature, salinity, splash pool size, and other morphometric parameters of the splash pools, seawater depth, and also like the different community of the organisms. And what we did is that we were very lucky because we collaborated with the Carmavi Biology Station in Curaçao. We could also like set up a lab, and our setup and inspection of samples looked like this before. And to our surprise, what we found, if we try to correlate all the parameters with the presence of multicellular colonies in our splash pool samples, is that salinity actually had a significant correlation with the presence of multicellular colonies or sheets. And indeed here in this graph, you can see salinity with a small subset of splash pools, and the multicellular colonies are found in the magenta points, meaning that mostly when salinity is in this permissive range of a bit like seawater salinity to maybe twice the seawater salinity, we could identify multicellular sheets, but when salinity is too high, we were not able to identify sheets or colonies. We also performed several follow-up experiments in which we were coming up to the field every day and trying to monitor the same splash pool over time. You can see here an example of a splash pool that undergoes evaporation until complete desiccation and then eventually refilling by potentially rain and waves. And if we examine the presence of multicellular colonies in each of the samples, we can see that both in the initial splash pool sample, there were colonies. Then upon salinity increase or evaporation, colonies disappeared completely. And then upon refilling, we were able to find colonies again. The question was, what happened to the dry splash pool? Because there's the possibility that maybe the organism is coming from the rain or maybe from the sea, but alternatively, maybe they never left this splash pool environment or this splash pool context, meaning that there was a potentially desiccation-resistant form. And for this, we did this very cool experiment in which we sampled soil samples and we rehydrated them in the lab. And to our surprise, we could recover multicellular colonies from the soil samples that were rehydrated in the lab. So this actually suggests that the cycle of this organism in the field is highly correlated with the dynamics of the splash pools, but we actually wanted to find a more reproducible conditions in the lab, trying to see if we can mimic what happens in the environment in the lab to verify our results. And for this, we built a very simplified model of splash pool evaporation based on our empirical observation. And we used like a very simple setup using an incubator, trying to match the gradual evaporation rate that we found in the field. So you can see here normalized salinity across more than 12 splash pools that we monitor in color. And then in our black dots is the evaporation rate that we matched in our experimental setup. And it looks like this, so nothing fancy. And indeed, we could reproduce loss of multicellularity upon gradual salinity increase in the lab. So you can see that we start with normal salinity conditions, we start the evaporation process, colonies, they start to dissociate, they start to look funny, and then they completely lose multicellularity. If we rehydrate, we can restore multicellularity, and this confirms that this evaporation and rehydration cycles control these transitions into and out multicellularity. And for the sake of time, I'm not going to discuss further what happens to this unicellular stage or desiccation-resistant stage that we call cysts. But basically, cells undergo cell growth arrest and some cellular remodeling, losing some features to resist desiccation. And both in the field and in the lab, we could confirm that this can happen for a long period of time. So they can remain resistant to desiccation for over a week. So basically, just to sum up this part, we've seen that unicellularity and multicellularity are controlled by these transitions of lower salinity and higher salinity in the splash pool cycles. We've seen that low salinity basically favors multicellularity, and we know that there's a feeding and a growth advantage along permissive salinity range. Clonality and aggregation are both allowed, so C. flexa can develop multicellularity both using clonal division, using aggregation, or a combination of both processes. Whereas if we go towards a higher salinity or until desiccation completely, there's a loss of multicellularity, unicellularity is favored, and in this case, transition to cysts confers a survival advantage. And of course, not all the cells, they can resist desiccation, so we have massive cell death as well. What is cool here is that there's a trade-off between aggregation and clonality, and the fact that C. flexa has this mixed mode of multicellularity allows for fast establishment of multicellularity in a wide range of conditions, especially because medium-high salinity completely inhibits clonality, but aggregation is still allowed, meaning that it has the choice to become multicellular at different salinity conditions or in a different salinity range using multiple strategies. I'm not sure if I'm doing well on time. I have like one last part. If you think, Dinah, that it's a bit short, I can skip it and go to the end, or I can just quickly finalize this part. So let me know, because I know that I've been like 18 minutes-ish. Otherwise, I can just quickly go over it.
You've got a couple minutes.
Okay, I will try to give a bit of an overview, and otherwise I can cut and it can be followed for questions later. So the last part that we wanted to ask is whether this clonal aggregative spectrum is actually plausible in the natural context, because just as a reminder, we validated this in the lab when we brought our samples and established the culture. And there are two main features that define the multicellular capacity of aggregation in the field. One is to have a sufficient cell density to be able to aggregate, and the other is to actually develop these kin recognition or self-recognition mechanisms to confer the ability to recognize each other and to selectively adhere and form multicellular bodies. So to examine this, what we did is that we isolated different strains from different regions. So we actually had 3 different strains, 2 that are coming from very close by neighboring splash pools that are separated for a few meters, and then another one that is separated for like a bit more than that half a kilometer. And we generated whole genome sequencing and stable cultures to compare their preferential aggregation capacities. So the whole genome sequencing was performed in collaboration with Jacob Stingwick at UC Berkeley, and also thanks to the amazing work that was done by Uzuki Horo. And then we also wanted to perform these preferential aggregation assays to see whether different strains could actually identify and select each other for forming colonies. So if we actually perform a very similar aggregation experiment as before, and we stain or label different strains coming from different splash pools and we mix them, we can have 2 potential outcomes that are different from the previous one. If they mix completely, it means that basically their genetic distance and their capacity of detecting each other is not in pro or favor in kin recognition. Whereas if they have different preferential aggregation capacities, that would be an indicative of potential kin recognition. So if we just mix the same strain with itself as a control, we can see that they mix perfectly and we have chimeric colonies. If we mix strain 1 and strain 3, we also can see that they can form chimeric colonies of 2 colors, indicating that they don't have kin recognition or that potentially genetically they are very close together. But our surprise was when we started mixing strain 1 and strain 2, in which we could see that there was pretty much segregation and different preferential aggregation. So colonies were mostly of one or the other color. And if we actually compare the genomes of these 2 strains and we look for those sites that actually exhibit the highest ratio of synonymous versus non-synonymous mutations, so looking for those highly polymorphic sites, those analyses were done by the amazing Uzuki and our collaborator Jacob, we could find some potential candidates that we hope we can validate in the future that are indeed involved in selective adhesion genes. So, for example, we have some cadherins, and also we have some other proteins enriched with immunoglobulin-like domains, indicating that potentially they evolve these mechanisms to have these preferential aggregation capacities and identify each other from different strains. So also, moreover, if we go a bit further and we mix different species, so here we are mixing C. flexa in pink and then a completely different species, S. rosetta, in green that develops clonally, we can see that indeed they don't mix at all. So at least kin recognition constrains aggregation both between different strains of C. flexa as well as different species. And I'm going to end here. Just as a small recap, I've shown you that flagellate cells of C. flexa, they have this mixed mode of multicellularity in which they can form colonies by aggregation or clonal division, that the life cycle or the transitions of the splash pool natural environment influence these transitions into an out multicellularity, and that normally high salinity induces loss of multicellularity and differentiation into these desiccation-resistant cells, and that there are like different selective advantages depending on whether you are at a low salinity, in which both clonality and aggregation are favored, and also growth and feeding, versus high salinity, in which unicellularity is favored and you have enhanced survival. And I'm going to end here. I just want to thank all my collaborators and the lab for the opportunity. Special thanks to Jose Andrés Rivera, which has been the co-author and collaborator of the work, and as well as also our collaborators at the Curaçao Biology Station. And of course, all of you for all your attention. I'll be super happy to take questions. Thanks a lot.
All right, so I'm going to be talking about the evolution of somatic differentiation. And specifically, I'm going to be talking about how somatic differentiation can evolve rapidly and repeatedly through changes in developmental plasticity. So the evolution of complex multicellularity is the transition from unicellular to differentiated multicellular organization. And this is an evolutionary transition in individuality. Selection is changing from acting on the level of the cell to acting on the level of the multicellular organism. The evolution of cellular differentiation is a key step mediating this transition. Now, cellular differentiation is environmentally regulated in unicellular organisms, like in choanoflagellates. Cells can change their state in response to the environment. But it's developmentally regulated in differentiated multicellular organisms. So what mediates the transition from the environmental regulation of cellular state to the developmental regulation of cellular state? Well, one possibility is plasticity. That is, changes in cellular state in response to the environment. But did the modification of plastic responses shape the evolution of multicellularity? And can new cell types even originate as a plastic response to the environment, shown here, that's later stabilized by changes in plasticity, such as the loss of plasticity, and then were further refined by adaptive evolution? And if so, what are the underlying mechanistic changes? So to address this, I use the Volvocene algae as a model system. They're a great model system for studying multicellularity because the clade contains unicellular organisms like Chlamydomonas, undifferentiated multicellular organisms like Gonium, or this Eutorina colony, where each cell is undifferentiated. Each cell is going to divide and give rise to the next generation, to a new multicellular organism. Then you have species like Pleiadorina and Volvox. These species have specialized somatic cells. Those are these little cells that you see here and here. And species like Volvox also have specialized germ cells. The cellular differentiation has been well characterized in the Volvocene algae, and there have been repeated gains and/or losses of somatic cells. If you take a look at this phylogeny here, you don't need to read any of the names, just take a look at the colors. So everything that's in teal is undifferentiated. Everything that's in yellow or purple has somatic cells. So you see that somatic cells evolved here in this lineage leading to Astrophominine, and here in this Volvox lineage. And then I want to draw your attention to this smaller clade here. This is called the EVP clade, Uterina, Platorina, and Volvox. So it consists of three genera, and each genus has evolved repeatedly. So we see Uterina in teal evolving repeatedly. We see multiple instances of the evolution of Platorina in yellow and Volvox in purple. So I'm going to focus on a Uterina species that's an outgroup to the rest of the EVP clade, shown by this arrow here. And I previously found that Uterina can develop plastic somatic cells following cold shock. So if you take a Uterina colony population and you subject it to 2 hours of cold shock, you'll see that some cells are going to differentiate into somatic cells. And this is regulated by both cell responses to the environment and to organismal developmental changes. And this is a plastic response. It does not persist past 2 generations. So, did somatic cells evolve through changes in plasticity? Is this plastic response to cold shock that we see modified during the evolution of obligate somatic specialization? So to test this, I've been using experimental evolution. And I've got two sets of experiments. In the first one, I have several lab lines. So I started lines from a single Uterina colony. I subjected it to 2 hours of cold shock every 5 days, which corresponds to 2 to 3 generations. And I had 2 seasons of repeated cold exposure, one with 5 rounds of cold shock and one 7 months later with another 7 rounds of cold shock. We assessed phenotypes months after the last time they experienced cold shock, and we subcloned and sequenced an experimentally evolved line and sequenced a control. I also have more than 800 introductory biology students at the University of Arizona working in teams as part of a course-based undergraduate research experience that's created and run by Ryan Rubiones. So each student team has a control line, and they have a line that they're experimentally evolving. And each team is assigned one of 3 treatments. These treatments differ in the number of rounds of cold shock and the duration of time between instances of cold shock. So students are subjecting the lines to 2 hours of cold shock every 3 to 4 days or every 7 days. And these 3 treatments have either 5 or 10 rounds of selection. At the end, students imaged 50 colonies per line, more than 3 generations after the last time they were cold shocked. And then I assessed the phenotypes with an image analysis pipeline and manual curation. So what did I find? First, let's look at the results for the lab lines. On the y-axis here, I'm looking at the proportion of differentiated colonies, and I'm calling this population level differentiation. If you look at the controls, you can see that the pooled controls have less than 20% of colonies that have one or more somatic cells, and the 2 control lines shown by these 2 separate dots are very similar. If we look at the experimentally evolved lines, we see that they have significantly more colonies with somatic cells than do the controls, and the 2 experimentally evolved lines are not different from each other. Additionally, I pulled out some single colonies from my experimentally evolved lines and I looked at their descendants to see how many differentiated colonies those descendants had. And I identified one lineage, which I'm calling 11A5, has basically obligate somatic cells. About 95% of colonies have very clear differentiation. And you can see some images of 11A5 here. Now let's take a look at the lines from the cure. So we can see that for each treatment, the experimentally evolved lines evolved a significantly higher proportion of colonies with soma than did the controls. Now, each one of these little circles here corresponds to a single student team's line. You can see that some of these circles have bold outlines and some don't. So every circle with bold outline is a student team where population level differentiation differed significantly from the controls. In treatment one, 11 of the 21 lines evolved significant changes in population level differentiation. Treatment two, we see that 3 of 10 lines evolved. And in treatment three, 5 of our 15 lines evolved changes in population level differentiation. And on the side here, you can see some examples of differentiated colonies from example teams from each treatments. You can really clearly see these small somatic cells. Now, not only did the proportion of colonies with somatic cells evolve, but I also saw changes in the proportion of somatic cells per differentiated colony. So I looked to see whether these two are correlated. Now, on this figure here, on the x-axis, we've got population level differentiation. That's the proportion of colonies with soma. On the y-axis, we have colony level differentiation. This is the proportion of differentiated colonies that have, or the proportion of somatic cells per differentiated colony. And you can see that there's no relationship between the two for the controls. But in the experimentally evolved line, these two are significantly correlated. That is, lines that have higher population level differentiation also have higher colony level differentiation. And this relationship is stronger if we only consider lines in which population level differentiation has evolved. So in these lines, we're seeing this correlation between the proportion of colonies with soma and the proportion of soma per differentiated colony. But this relationship does not exist in lines in which population level differentiation did not evolve. So then I look to see if there's any convergence happening with Pleiadorina. So I took a data set of 16 celled colonies of Pleiadorina starii that was published in Heron et al. 2014. And I compared the mean proportion of somatic cells per differentiated 16-celled colony for each line to that of Pleiadorina. So in this figure, on the x-axis, we've got population level differentiation again. On the y-axis, we've got colony level differentiation again. So it's for 16-celled colonies for all lines. And here's Pleiadorina starii. So all Pleiadorina starii colonies are differentiated. And on average, 25.5% of colonies, or 25.5% of cells in a colony are somatic. And I found that 3 of my experimentally evolved lines evolved more somatic cells than Pleiadorina. This is 11A5, and then 2 of my cure lines that are shown in these blue points here. And these larger circles around the points are those 95% confidence intervals. An additional 8 lines evolved more somatic cells than controls, but they did not have as many somatic cells as did Pleiadorina starii. So, or did not have more somatic cells than Pleiadorina starii. So these are all of these points in green here. Then all of the points in yellow correspond to lines that did not evolve more somatic cells than controls. But these are just averages. So what does the distribution look like here? So I looked at the number of somatic cells per differentiated colony. That's what I've got here on the y-axis. And we can see for our controls The distribution is really peaking around 0 somatic cells, and some of them have one. Same is true for our pooled lines in which the number of somatic cells did not evolve. For our pooled lab lines, we're starting to see the second peak. But let's focus on some of these lines that evolved more somatic cells than plate arena. So these are the ones in blue here. So if we take a look at 11A5, we can see that this distribution of somatic cell number has really changed. We've got this second peak in somatic cell number that we're not even seeing among the 16-celled plate arena starry colonies, which are fairly constrained. Additionally, in some of these lines, there's what looks to be a loss of constraints in somatic cell number. We see that happening here, and we're seeing substantial changes in this line as well, even though there aren't any changes in the mean number of somatic cells relative to plate arena. So it was a lot of data. Let's go ahead and summarize that. So again, in my controls, on average, about 16-17% of colonies have one or more somatic cells, and about 17% of cells in a differentiated colony are somatic. In an 8-celled colony, that corresponds to there just being one somatic cell. In the experimental lines that didn't evolve any changes in cellular differentiation, we're seeing an average of 26.7% of colonies that are differentiated and 20.7% of somatic cells per differentiated colony. So there is a slight increase in the number of somatic cells per colony, even though it isn't significant. In our experimental lines that did evolve changes in population level differentiation, see that more than half of colonies have one or more somatic cells, and a quarter of cells in a colony are differentiated. And in 11A5, almost all of the colonies are differentiated, and about 40% on average, of cells in a colony are somatic, although we see that there's a really wide distribution there. And here you can see some additional images of experimentally evolved uterinas. And I've compared them to different Platorina species here. So Platorina is an obligately differentiated genus that has evolved repeatedly. And somatic cell percentages range from 12.5 to 88%. But most Platorina species have between about 20 and 50% soma per colony, with a lot of them falling in this 25 to 40% range. If this was to be an 8-celled colony, that would correspond to 3 to 4 somatic cells per colony, which is again very similar to what we see in these experimentally evolved lines, especially the ones that converged with Platorina starrii. But what are the genetic mechanisms underlying these changes? What is mechanistically changing here? So I used long read sequencing, and I looked at the genomic changes in 11A5 relative to our reference and to the control line 1A. I identified 18 annotated genes that were mutated only in 11A5, did not have any mutations in the coding sequence or in a regulatory region in the controls. Of these, about 94% had at least one mutation in a regulatory region. I also identified 14 genes where I could find homologs in obligately differentiated volvox, in obligately differentiated astrophamini, and in unicellular chlamydomonas. In 1A, I found 31 annotated genes that were mutated. 1A wasn't subcloned as recently, so there's more genetic variation in it. 87% of these genes had at least one mutation in a regulatory region, and I identified 24 genes with homologs in these three species. Then I used existing data sets to look at soma-specific expression of homologs in Astraphomini and Volvox, and to look at cold-induced expression in Chlamydomonas. This sequencing was all done at Coast Genomics. So what did I find? Found that significantly more 11A5 genes had homologs that had both cold-induced expression and soma-specific expression. So in 11A5, 71.4% of my genes had homologs where there's um, cold-induced expression in Chlamydomonas and soma-specific expression, or cold-induced expression in Chlamydomonas and soma-specific expression in Astraphomini, Volvox, or both. In my control 1A, only 30% of genes had homologs that have both cold-induced and soma-specific expression. So what is happening here is that you've got obligate differentiations evolving through the modification of genes whose homologs are affecting both this cold shock response and affecting somatic differentiation. So it's genes that are lying in the intersection of these cold shock response and cellular differentiation modules. Then I took my 11A5 genes and I mapped them onto the known cold shock response pathway that's laid out in Armilova 2020. This is how summarizes how Chlamydomonas and plants respond to cold stress. And I found that there's changes in all of these key stages of this cold shock response pathway. See changes in signaling, genes affecting signaling molecules, affecting regulatory factors, affecting transcriptional regulation. And then downstream of transcriptional regulation, I see that there's a lot of genes involved in protective processes and in cell cycle regulation. Additionally, identified two genes that are involved in cell wall modification, although this is not part of the canonical cold stress response pathway. Well, let's dive into the identities of these genes to figure out what's going on a little bit better here. So all of the genes in blue have at least one Volvox or Astraphomini homolog soma-specific expression. Everything in italics has a Chlamydomonas homolog that's expressed following cold shock. And I've also included genes for which I could not identify Volvox, Astraphomini, or Chlamydomonas homologs. And I really want to draw your attention to this gene in this box here. This is gene 603. This is a transcription factor, likely a transcription factor that has a squamosal binding protein-like domain and a BTB/pause domain. So squamosal binding proteins are developmental regulators that affect the transition from reproductive to vegetative developments. And BTB/pause domains often bind repressor complexes. Now, the homolog of 603 is downregulated in Chlamydomonas in response to cold shock. And this gene has a multi-exon deletion, so it's lost its SVP DNA binding domain. So what's happening here is we've got this transcription factor that's usually downregulated in response to cold shock that might act as a repressor and might regulate the transition between reproductive and vegetative growth. And this transcription factor is no longer functional. This might also explain why we're seeing so many changes that are happening, regulatory changes happening downstream of this transcription factor. So let's bring this together. So I've showed you today that somatic differentiation can evolve really rapidly and repeatedly through the modification of developmental plasticity. I showed you that Uterina develops plastic somatic cells in response to cold stress. I showed you that repeated cold stress causes the rapid evolution of obligate somatic cells. So that's the stabilization or modification of this plastic response. And I showed you that the proportion of somatic cells per colony also evolves really rapidly through evolutionary refinements. This is driven by selection that favors somatic cell development. I don't have time to show that to you today. And this is likely occurring through the modification of genes that might be in both a cold response module and in a somatic differentiation module. So, what do I think is happening during the evolution of somatic specialization? So, if we think about uterine development, it starts from a single cell in a colony. The cell is going to divide like the other cells, undergo inversion, turn into a little daughter colony that's going to hatch out and form the next generation. And we can summarize this as going from a single cell to a multicellular colony. And in control conditions, development typically results in an undifferentiated colony, though sometimes we do see the development of differentiated colonies like this one here. And we can further visualize this or represent this as a ball rolling down a hill. This is Waddington's epigenetic landscape, and this is just a conceptual developmental landscape. Now, cold shock changes the shape of this landscape so that these lines that are cold shocked are likely to proceed along either of these developmental pathways because cold shock has modified the shape of the developmental landscape. Now, in my experimentally evolved lines that were repeatedly cold shocked, this developmental landscape that was previously induced by cold shock has been stabilized, such that development now consistently proceeds along either of these pathways. And in lineages like 11A5, we see that differentiation is now consistently occurring along the pathway, resulting in obligate differentiation, similar to that of Pleiadarena. So I think that developmental landscapes are being modified during the transition to obligate somatic differentiation. And with that, I'd like to thank the Michaud Lab at the University of Arizona, especially Rick Michaud and Yongwon Yu, who did the preliminary phenotyping. To thank the Olson Lab at Kansas State University and Augusta University, actually Brad Olson. And Lucas Williams has done some of the AlphaFold gene models here. And a major thanks to Ryan Rubiones, who has run the cure at the University of Arizona. And I will be happy to take any questions you may have. All right, so I've pulled up the chat here.